And we have gotten to the exciting gene therapy portion of the talk. And it's my honor to introduce Dr. Matt Fuller from Ultragenyx. And he's going to teach us about gene therapy and what it might mean for FAM.
Hi, I really appreciate the opportunity to be here and chat with everyone about gene therapy and hopefully offer a balanced view of what is possible. I will say just in the beginning that given the size of your gene, I think there's a lot of hope for developing therapies of the genetic type for this disease. But as Ethan just suggested, there's also a lot of other things. And what I like to tell all the different family foundations that I talk to is never put all of your eggs in one basket. Because just because gene therapy seems like a viable option, or maybe a small molecule or anything like that, it's always good to have multiple things going because you never know when something might hit a roadblock. And so that's usually my number one advice for them. So what I'm going to do today is try to briefly walk you through gene therapy and some of the things you'll want to think about as you develop gene therapies or think about developing gene therapies for FAM. And just note on the bottom left, I do have my email address here. As many people have said, a lot of this content is new to people. And so if I can help by explaining this or other types of scientific concepts, I'm happy to do that. So just reach out. Okay, so let me make this smaller. Sorry, I have to make the little people smaller. So the first thing we need to do to really know if we can do gene therapy is just understand what are we talking about. And so if you were to take one of your cells out of your body, you would see that you have a nucleus that's running the cell. And within that, your DNA is all tightened up into a chromosome. If you were to magnify that a bit, you would see that it forms a structure with these proteins called histones that basically help wrap it around so you can compact it. But more importantly, if you zoom in on that, that's when you see something called a gene. And a gene is a basic genetic unit that encodes a function. And so a gene is located in a specific section of DNA on a specific chromosome. In the case of FAM177A1, it's located on chromosome 14, Q13.2, which I believe in the very beginning, Charlie gave an explanation sort of what that means. And he also gave the explanation that genes are encoded by the DNA nucleotide alphabet of A, G, T, and C. And it's important to understand that genes encode overt functions that are typically coded for proteins. But genes also have lots of other things that they can do. But what we're going to talk about today is genes encoding overt functions, specifically for FAM. And it's important that if you zoom in on a gene, that the nucleotides are the things that have the mutations or that have deletions. And that's really what we need to understand at this level to be able to do gene therapy. It's also important for all of us to know that alterations in genes can, but do not always cause dysfunction. So we're here today because we're all affected by people that have some sort of disease caused by mutations or alterations in nucleotides. But everyone sitting on this call also has a variety of mutations that are not affecting us at all. And these are silent mutations. And so it's just important to know that some, but not all, do cause dysfunction. So what exactly does DNA do? DNA encodes for about 20,000 genes that have discrete cellular functions. And these are used by our cells and bodies for our day-to-day existence. And these functions are facilitated by pathways that utilize sort of the diagram I have here on the left, where you have DNA that's made into mRNA, and then that's made into protein. And proteins are the things that have the functions that carry out our day-to-day tasks, vital just for us to live and survive. And some of these examples include processing metabolites, muscle contraction, DNA replication, moving other proteins around, activating or inhibiting other proteins. And as we heard earlier from Ugur, we heard that FAM probably has something to do with interacting with the Golgi and maybe something to do with the stress response. And so that's something that, this entire organization will need to work to figure out. Because understanding that will be key to making any sort of therapy that we're going to develop functional. And it was important for me to sort of explain all of this to you, because what you need to know is that gene therapy seeks to replace or repair these very specific pathways with molecules such as the gene itself or the DNA itself, when the typical function is not being provided due to a mutation. So we are actually looking to replace this part right here, so we can make the appropriate mRNA and we can make the appropriate protein. And there's a variety of ways that we can go about doing that. So just the most basic definition of gene therapy that we can have is that the treatment or prevention of a genetic disease by introducing genetic material expected to provide a necessary function. And sort of just thinking back to what I showed you earlier, you have a gene, we either replace or repair that gene in some manner, and then you have the functional protein that you're looking to replace. Okay, so there are a variety of ways that you can go about thinking of gene therapy, and I'm going to talk to you today about sort of the three most basic, but if we were to get into the details, there's many, many ways of approaching this. So importantly, gene therapy approaches include the delivery of a package or a cargo. And when you deliver this cargo, you design them to do one of these three things. You're either trying to add back a functional or wild type copy of a mutated or missing gene causing disease. And I have a note here at the bottom, just in case people throughout, their sort of tenure growing into science hear the term wild type, that refers to a natural or common version of a specific type of protein or DNA and things like that. So you might hear wild type thrown around. It just means the normal version. You can also seek to inhibit, inactivate, or knock out a mutated or overexpressed gene that's functioning improperly. This is probably not the case of what's going to happen with FAM. You can also edit a mutated gene back to a functional copy or a wild type copy of that gene. So these are the three approaches. I think A and C are probably the most likely approaches that would be utilized to treat FAM. So how do you go about actually developing a treatment with gene therapy? There are three basic tools that people utilize right now. One of them is adeno-associated virus, which you can see over here on the right. And that is a small virus that is both a delivery vehicle and the cargo. So the delivery vehicle is this protein shell from the virus that when you inject it into a person finds its way to a target cell. And then this DNA that you see here actually encodes for the protein that you're trying to deliver to the person. Another common tool used is a lentivirus. And it's very similar to the AAV in this concept that you have a protein shell, it finds its way to a target cell, and then it delivers the cargo. There are some big differences between AAV and lentivirus that we can talk about, but it's probably not important to talk about here. And then the last one, and the newest and probably most exciting, and also the one that we read about in the New York Times, is gene editing. And so that is specifically a cargo, but that has to be coupled with a delivery vehicle. And that is one of the most complicated parts of this technology right now, because it's so large, trying to figure out the best way to get it to the proper target cells. And also, this is a new technology. But what's important to note is about 2 years ago, I think that's about when I first met Jill, I told everyone that, gene editing right now is just not really a thing that's going to be tractable for people. And 2 years later, as I'll mention later, Vertex and CRISPR Therapeutics have something that they're trying to get approved by the FDA. So research moves fast, and therapies can move fast when people are really putting all their effort into it. And that's important for all of us to just keep up with what's going on. Okay, so just to kind of compare and contrast quickly AAV and lentivirus, why would you use AAV? It's good for in vivo delivery, which means delivering it directly into a patient. It's ideal for gene replacement. And another good thing about it is that there's academic and industry manufacturing experience. People have been utilizing this for 20, 30 years. And so they know how to make it. They know how to work with it. But what's difficult about it is that it's a very small virus. So it can only hold about 5,000 bases inside of it before it's just completely full. Now, I'll show you later, one of the things that's good about FAM is that it's small. And so it should be amenable to using AAV. And that's a positive here. But Another challenge is that it's incredibly expensive to manufacture. And I have some slides on this later. But some of the diseases that have been approved, that have approved treatments on AAV, the doses are like $2 million. So it's incredibly expensive, even if you have an approved treatment. For lentivirus, this is good for what's called ex vivo delivery. Or what you do is you remove patient cells, the target cells, you treat them with this virus, and then you return those cells to patients. My opinion is this is really not what you would do for FAM. So I don't really know that it's worth kind of going into this much more, other than it does hold more cargo, so that's a positive for lentivirus. One of the disadvantages, though, is that it does have a higher integration and genotoxic risk. So you do need a good reason to utilize this. Yeah, and whoever put this in the chat, this is exactly it. It's the one I'm talking about for beta thalassemia and sickle cell anemia. Okay, and so on to CRISPR-Cas9. This can now be used for many things. So this was originally conceptualized as being used for gene knockout. And over the years, researchers have been able to add on different uses, including gene knock-in or replacement. So adding a gene in to your genome, base editing. So actually going into the specific part of your DNA that has a mutation and changing that back to what it should be. And then there's also a new modality within this called prime editing. And it's sort of a combination of base editing and gene replacement, in that you go to the very specific part that needs editing, but you can add sort of big chunks. And so you can sort of think of base editing as, you go in and you white something out and you put the right, letter back in, whereas prime editing, you do cut and paste and you put, a sentence in there. And so both of these are really powerful ways to manipulate your DNA to fix the gene with a really strong therapeutic effect. I will say right now, ex vivo applications are really the most successful ways this is being used. And the reason for that is it's difficult to deliver these because of their size all over the body. There's also concern of having off-target effects. If you don't target the exact part of the DNA, you can have problems. And then finally, the targeting strategy has to be tailored to specific DNA sequences. So again, if you think about the way I described it, if you have a specific piece of DNA and you have certain mutations you're trying to fix, but we know from the research you've seen earlier that not everyone has the same mutation, you potentially have to make drugs for every single mutation that exists. And so what has happened for other drugs, which I'll describe later, is they've taken the most common mutations, made a therapy for that. But the problem is that only covers, in that case, about 10% of patients. And so, all of these things you just have to sort of think about as you're developing these treatments. So what makes a good candidate for addition gene therapy? Does your transgene fit? So as I mentioned, for AAV, you have about 5,000 bases to use. I updated this, but it didn't make it in here. FAM actually is 711 base pairs. I had the wrong, what's the right word? I had the wrong isoform. So it's 711 base pairs. You need some additional sequences, including these sequences here, which allow for the packaging, and then this on signal and this off signal. But basically, this gene appears to be very amenable to gene therapy. But some other questions do have to be asked. Can you deliver this to the appropriate target cell? And is that target cell amenable to being corrected? And so those are things, though, that researchers will be able to understand, because as Ethan and others have shown, these models are becoming more and more sophisticated. Now, one of the things that I think does need to be figured out is whether or not there is a Goldilocks scenario, which is what we call it for a lot of different central nervous system diseases for FAM, in that sometimes you can have too much or too little of a protein and you have to be in just the right area. For some liver diseases where you're metabolizing something, you basically cannot have too much of a certain protein, so you're good. So this is just something that researchers will need to figure out. And what you can do is, if you find out that you are in a Goldilocks scenario, you can design your vector to, sort of remain in the right areas that you need them to be. Oh, so I updated it here, but not there. So yeah, you passed the first test. It's small enough even to consider a self-complementary design, which is something we can talk about later, but that's a special design of AAV that turns on faster. But we definitely need to understand better the models that you would use to understand the target cell types and the target expression levels you would have. So what makes a good candidate for inhibition gene therapy? It's basically, do you have a good target to inhibit? And it might come out at some point that there's some interaction with FAM that's important and you could inhibit that. But I think so far the disease methodology suggests that that's not the case. So this is probably not going to be a good case for inhibition gene therapy. But you do have the same concerns. Can you reach the right cells? Are those cells amenable to gene therapy? So it's kind of the same thing. Do you have a good target? Can you get to the right cells? Are those cells going to be amenable to what you're trying to do to them? And then finally, we have CRISPR editing. What makes a good candidate for CRISPR editing? And again, it's very similar. Do you have a good target? So you need a clearly definable editable region. And so if there are hotspots of mutation, that can be very helpful. And so the example I sort of foreshadowed earlier was Leber's congenital amaurosis. It's been targeted by Editas to a mutation in intron 26. That's the most common mutation because the gene was big enough they couldn't make a therapy over all the different mutations, or they might have to make 50 different drugs as recognized by the FDA. So what they did is they looked at the most common mutation. But again, as I mentioned, that only targets 10% of patients. And so you're leaving 90% of your patients behind with that one therapy. So that is something to think really long and hard about when you're developing this. And it could be a reason why if prime editing comes along, it will be a much better type of CRISPR editing. You also need to think about if your gene is truncated by mutation. So is it shortened or is it deleted? Because you have to have something to edit. And so it's just going to, again, it's going to depend on the type of genetic cause of the disease per patient. And then really the same to other issues. Can you reach the cells that you need to? And are those cells amenable? And you might be asking yourself, why do I keep asking these questions? It's important depending on the organ you're trying to get to. If you're trying to get to the liver, that's really easy. The liver is a sponge. It takes up everything. If you're trying to get to very specific cell types in the brain or in the spinal cord, that can be very difficult. And so you really need animal models to help you figure out if you're getting to the cells that are really causing disease. So potential CRISPR approaches and considerations for FAM, we really need to identify clinically relevant editable regions. And prime editing could be a good way for gene replacement or large corrections, especially because prime editing actually can insert 1 to 10 kilobases. And so you could theoretically insert the entire coding region, the 711 bases, somewhere into the genome for FAM if you wanted to do that rather than having AAV gene therapy. But what are the challenges? CRISPR, again, has only been successful in ex vivo treatments. And so we really have to have companies and academics get to where this is tractable for in vivo treatments. We don't know the number of cells that have to be targeted. For CNS diseases, you often have to reach 20, 30% of cells. It's not enough to hit 10% of cells. Whereas in some liver therapies, you can reach a really low number of cells because they're just turning over metabolites. And also base editing has not yet been used successfully in humans. So cutting something out or inhibiting something has been, but base editing is not. And so these are just things that you will have to keep your eyes on as an organization to see if this will become tractable in the future. But the advantage is obviously that you have the potential to directly fix the underlying genetic cause in the cell, and it will be permanent. Okay, so I know we're behind on time. I'm almost done here. I wanted to share with you sort of at the end the fact that there are gene therapy success stories. So although there are advantages and challenges for everything, and there's a lot to think about for developing these, other people have done this for their diseases. And so right now there are 4 approved AAV gene therapy products, although one was withdrawn in the EU. And this is for Leber's congenital amaurosis. This is for spinal muscular atrophy, and one that was just recently approved for hemophilia B. And I'll just highlight quickly the one for spinal muscular atrophy. If people are not familiar with this disease, this is an incredibly detrimental disease to young children that often die by the age of about 2.5 because they're unable to breathe and they're unable to move. And the children that have been treated with this AAV are now walking and breathing and beyond 2 years of age. And so, you know, when researchers can get these products right, when they're done right, they really can be life-changing. It's a lot of work to get to it. There are also quite a few approved retro and lenti products, but as I mentioned, that's really better for taking cells out, treating them, and putting them back. And finally, as was shared earlier, there has been something submitted to the FDA for approval for CRISPR products, and that's for beta thalassemia and sickle cell disease. So CRISPR is also moving forward as something that's going to be tractable in the future for generating therapies. So I think I've sort of talked about all of this, but I'll just really highlight the key to successful outcomes. You have to have solid basic clinical science. You have to have a good cell and target tissue choice. And the reason for this is if it's you or if you're trying to get a pharmaceutical company to develop these, they have to be able to have biomarkers and understanding of this disease to get it approved, to work on it and to get it approved. That's really important. It's also important for you to know the limitations of any of these gene therapies, which is that one, they're incredibly expensive to generate. As I mentioned, some of the doses are $2–3 million. And also there are at times safety concerns. And so I mentioned to you that Zolgensma, which treats spinal muscular atrophy, was life-changing for many of those children. But there were 2 children that died during the trial because they had high dose and it affected their heart. And so there are always safety concerns with any therapy that is being generated. And that's something that needs to be, you know, really well studied during these trials and during the preclinical work. Okay, so last slide, key takeaway. Gene therapy does have the potential to halt or treat diseases with a one-time treatment, and there are successful FDA-approved examples. Success, though, is critically dependent upon solid basic science, understanding the cell and tissue target choice, and the ability to make the vector. And I would just also say that whoever starts working on a therapy, make sure early on you can make whatever you're making, because if you have something and you find out 3 years into it, you can't manufacture it, it's not really that helpful. Each gene therapy strategy has pros and cons, and you just really have to think long and hard about which one is best. Or it can be that gene therapy is not the best thing, and maybe a small molecule is better. So as I mentioned in the beginning, really have multiple horses in the race, and just try to spread out your risk as much as possible. And finally, manufacturing and reimbursement are expensive. And so hopefully that will come down over time, but it's just something that everyone needs to be aware of. And with that, I'm done. I'm happy to take any questions. And also, if you want to email me, I'm happy to answer questions that way as well.